Hi, my name is Michelle Cote, and I am an upcoming senior in biomedical engineering. I'm here to talk to you today about my summer research through the SURE program in the Micro, Nano, and Molecular Biotechnology Lab of Dr. Takayama under current graduate student Andrea Jovic. The title of my presentation is Visualizing Calcium Dynamics in Non-excitable Cells under Pulsatile Stimulation with Fluorescent Microscopy. So first, a little bit of background on the research that I'm conducting. Non-excitable cells respond to chemical and not electrical stimulation. An example of an excitable cell would be a cardiac cell in your heart that responds to electrical stimulation from surrounding neurons. Physiologically speaking, signal transduction mechanisms in non-excitable cells respond to pulsatile stimulation as opposed to continuous stimulation. So for example, after you eat a large meal and your blood glucose is high, insulin is secreted from pancreatic cells in pulses instead of it flooding through your system. And intracellular calcium signaling is an important regulatory mechanism for these cellular processes. So for example, calcium oscillations lead to the insulin secretion from your pancreatic cells, and any dysfunction in the insulin release is linked to type 2 diabetes. So the current research goals for our project is to use microfluidic devices to stimulate non-excitable cells with pulsatile exposure to a ligand, and to visualize the calcium response using fluorescent microscopy. We then analyze the cellular response patterns to varying stimulation conditions. Under our current setup, we can vary 3 parameters, which you see in this figure here. S stands for stimulation duration, which is how long the cell is exposed to the ligand. R, the rest period, which means how long the cell is allowed to rest from being stimulated with the ligand. And C, the concentration of the ligand itself. The ligand that we use is carbachol, a stable derivative of acetylcholine, which binds to the M3 muscarinic receptor, which is implicated in the insulin secretion pathway. Previously established mathematical models have shown certain phase lock rhythms that predominate in these cellular calcium responses. So as you can see in this figure, for every carbachol input, which are these black rectangles, the cell responds with a calcium spike. And as you can see here, the cell only spikes 1 every 2 pulses of carbachol. This we refer to as skipping, and it's a 2-to-1 cell signal to cell response ratio. So if you can imagine a cardiac cell in your heart that only responds to every other electrical input, this could lead to an AV block and possibly a cardiac arrest. So using this mathematical modeling, we can predict the signal to response outputs and the predominant responses that occur if we hold constant 2 of the 3 parameters. So here you can see that the 2-to-1 and 1-to-1 signal responses are predominantly found. The experimental setup that we use to study these calcium oscillations is a microfluidic chip made from PDMS, an organic polymer. The chip has 2 microchannels, one from the left and one from the right, that come together in the middle to form a Y-shaped junction. In the Y-shaped junction is where we seed and culture the cells. Each channel is fed from one of 2 inlets, these 2 round, larger fluorescing circles, and we use a programmable Braille pumping device that moves Braille pins to pump the liquid from the left and the right. And using this program, we can time the pulses such that we pump the ligand from the left and the media from the right in order to create square wave pulses. We put the PDMS chip on the Braille device on the microscope, which you can see on the right, and then we hook up the Braille device to the computer and the camera. And using the camera, we can collect time-lapse fluorescent images. And if you look on the computer screen in the picture on the left, you can see splotches of yellow, red, and white, which corresponds to increasing intensity in the calcium concentration. From this, we can record and analyze the cellular calcium response. And this is some actual data that we have collected. So here we have plotted 2 cells, cell 1 and cell 2. And in the top cell, you can see that the calcium spike occurs every other, and in cell 2, the calcium response occurs for every pulse of ligand. So from these experiments, we have confirmed the efficacy of our microfluidic device at delivering square wave pulses. And in accordance with the established literature, the cellular calcium response falls into 2 main signal-to-response ratios, 2-to-1 and 1-to-1. And while holding the other two parameters constant, increasing the concentration of the ligand, the stimulation duration, or the rest period between stimulation increases the fidelity of the cellular response and allows us to see more one-to-one signal-to-response ratios. In the future, we hope to expand this search to other signal transduction mechanisms. The pathway that we use stimulated by carbachol is a G protein coupled receptor pathway, which involves many thresholds that need to be met in order for the signal to be passed on farther down the line. And another hormone that is in your body that is used for cell growth, proliferation, and metastasis is the epidermal growth factor, EGF, and it functions through a different signal transduction mechanism called a receptor tyrosine kinase, which doesn't involve many of the thresholds involved in the G protein coupled receptor. So we hope to study the cellular response to pulsatile stimulation with EGF to see if the phase lock rhythms that we see with carbachol are consistent across signaling mechanisms. That concludes my presentation, and I hope that you enjoyed it. Thank you.